On behalf of the Surgical Ethics Working Group, I would like to welcome everyone to our second annual Harvard Surgical Ethics Conference. Our first session this evening is Ethical Considerations in Pig to Human Xenotransplants. We will host 3 more sessions on September 12th, 19th, and 26th. Over 500 individuals have registered for the conference from around the world, and we would like to thank you for getting up early, staying up late, and taking time off from work or leisure to join us this evening. I believe we have an exciting hour and a half ahead of us. I would like to review a few housekeeping items before I introduce our topic for this evening and our 3 speakers. First of all, please submit any questions that you have during any of the talks using the Q&A feature found in the meeting controls at the bottom of your screen. Selected questions will be discussed after presentations. Please don't use the chat feature to send questions unless they're technical problems. The questions, we will try to get to all of them, but we may not be able to get to every specific question, but we will do our best. If you do have any technical issues, as I mentioned, use the chat feature to send a message to all the panelists, and a staff member will help. Also, please continue the conversation online with hashtag HMS Bioethics in your social media posts. If you're interested in other webinars, bioethics news, or HMS educational programs, subscribe to the Center's biweekly newsletter at bioethics.hms.harvard.edu/subscribe. Dr. Jay Fishman is a professor of medicine at Harvard Medical School, director of the Transplant Infectious Disease and Compromised Host Program at Massachusetts General Hospital. He is also the associate director of the MGH Transplant Program. He was also recently elected president-elect of the International Xenograft Association, a great honor. He is without question the content expert and voice regarding the risk of infectious disease transmission from animals to humans after xenotransplantation. Dr. Fishman will speak on the infectious risk and the challenge of safe xenotransplantation. Dr. Fishman?
Thanks, Doug, and we'll get into our slides, and thanks to the organizers for allowing me to be included in this session. I'm not yet seeing my slides, but let me start. There we go. Next, please. I have no conflicts that are essential for this talk. Next, please. So why are we talking about xenotransplantation? Why are we talking about putting organs from other species into humans? And the answer is fairly simple. We have a shortage of organs for transplantation as a cure of terminal organ failure. So in the United States alone, about 7,000 people died waiting for an organ on the list, and many more people don't get onto the list that could potentially benefit from such transplants. And of course, this is a worldwide problem. Next slide, please, which is affecting many of us. So as clinicians, much of what we talk about is how do we approach the patient with organ failure? Certainly better disease prevention. We have some new treatments, gene therapies such as CRISPR are coming into the fore now. We have many new stem cell therapies, particularly in oncology. We have improved human organ transplantation, and survival, even in the time that I've been working in this field, has more than doubled for organ recipients. Artificial organs have always been the dream of the future, but in fact, they haven't been recognized yet as a solution to our organ shortage. So animal organs have been a very popular solution in the press, but because of immunologic and metabolic barriers, have not been rolled out into clinical practice. Next, please. So pigs have become the donor species of choice. The easiest animal from which to transplant would be a primate species, a non-human primate, but because of infectious disease concerns primarily and ethical concerns, primates were very quickly discarded as potentials for organ transplantation. Swine have been studied for many years, certainly as farm animals, and they have breeding characteristics that are well described for commercial use. They can be sized to humans, they can be manipulated genetically, originally just with inbreeding and now using CRISPR-Cas9 and other gene manipulation therapies. And we can derive new strains of swine with desired characteristics, as I'll talk about shortly. There is a whole genome sequence available of the domestic swine, and some important viruses have also been sequenced from this particular species. Interestingly, swine cells are resistant to HIV, hepatitis B, and hepatitis C, which provides an advantage, particularly in regards to liver transplantation. The disadvantages are pretty great. All humans carry preformed natural antibodies to an alpha-gal sugar, which provokes hyperacute rejection, loss of the organ within 5 minutes after implantation. There are certain metabolic incompatibilities and certainly histoincompatibility for humans. Next, please. So people have been talking about using swine as organ donors for humans for many, many years. In fact, going back into the 1600s. The first baboon liver that was transplanted into humans was done around 1969. But I worked with David Sachs in the early 1990s on the infectious disease aspects of swine to potentially human transplantation. We cloned the porcine endogenous retrovirus, which may have been a great technical feat, but actually slowed the field down quite a bit in the years that followed. Next, please. What are the barriers of pig to human xenotransplantation? Well, certainly, as I mentioned, with the alpha-gal sugars, you can get hyperacute rejection within minutes to hours, preformed antibody in all human circulation, complement deposition, clot formation, endothelial injury, which generally would destroy an organ quickly. There may be initial xenograft dysfunction also in the early phase, which is metabolic, immune, and perhaps related to ischemia reperfusion of the transplanted organ. Delayed xenograft rejection occurs over days to weeks as preformed antibodies rebound and new antibodies are formed, and then chronic rejection over weeks as elicited T-cell immunity occurs with dysregulated coagulation. So there are many barriers to successful pig-to-human xenotransplantation. Next, please. This was addressed by Mohammed Mohideen and his colleagues, but in many centers now, by trying to do two types of approaches. One is genetic manipulation of the source animal. There are gene knockouts, including that alpha-gal sugar that I talked about, growth hormone knockouts to prevent overgrowth of transplanted organs, and then a variety of human transgenes. The human transgenes are for complement regulation, coagulation regulation, and inflammation regulation, and the placement of human genes within the pigs and expression in the pig organ prevents acute rejection and injury by the innate and complement systems of the human recipient. In addition, there is an intensive regimen of immunosuppression, so B cell depletion, T cell depletion, complement depletion, co-stimulatory blockade, and then on top of that, maintenance immunosuppression with a variety of T and B cell agents, anti-inflammatory agents, and co-stimulatory blockade. In that setting, with intensive immunosuppression and the genetic manipulation, prolonged survival of hearts and kidneys has been seen in baboon preclinical studies. Next, please. The infectious disease concerns emerged rather early. The concern was, of course, the implantation of pig organisms to which we did not have pre-existing immunity into the human recipients, so-called zoonoses. We really didn't have microbiologic surveillance or exclusion criteria for pig production or ways of monitoring recipients and their contacts for microbiologic safety. We didn't know which organisms to expect and what those organisms might do in a human host. How do we handle previously unknown organisms? And do the transgenes themselves have an impact on infectious risk? So for each of the potential organisms, we need diagnostic assays that are validated for use in pigs for breeding and humans for infectious disease monitoring. We need to monitor the recipient. How long? For their lifetime, for the lifetime of the graft? Do we have to monitor their social and sexual contacts? And is it possible that we could share these data so that there was enhanced safety across all the centers that were doing xenotransplantation? Do the new immunosuppressive regimens, these intensive immunosuppressive regimens, place them at risk not only for infection from the pig, but a greater risk for their intrinsic infections, just as we see in human to human transplantation? And how do we handle infection control? Who do we screen? What samples do we store? And what are the unknowns in this particular equation? Next, please. It's not all that complicated. The question is simple. Can organisms derived from pigs set up shop and cause significant infection in human recipients? Next, please. Interestingly, there was a recent report which demonstrated the bulk of confirmed human cases of swine flu have been connected to agricultural showcases across the United States. Now, you will be aware that the recent COVID pandemic was also connected to a place where animals were in close contact with humans for prolonged periods of time. So this is not a new observation, but there is concern about the spread of pig pathogens to humans. Next, please. So the goal is to make xenotransplantation as safe as allotransplantation relative to infectious risk, to develop assays, to characterize the biology of potential pathogens, and to understand the science underpinning all of these infectious risks. Next, please. Years ago, I set up a framework for categories of potential human pathogens as a way of thinking about this challenge. For example, there are many pathogens that are common for immunosuppressed human transplant recipients, cytomegalovirus, Epstein-Barr virus, and the like. For those types of pathogens, we have specific tests and assays, and for most of them, fairly effective therapies. For traditional zoonoses, things that are spread from pig to human, such as toxoplasmosis, there are also specific assays available and therapies. We don't know as much about species-specific agents, such as porcine cytomegalovirus, or porcine lymphotropic virus, or porcine circovirus. These are generally organisms thought to be incapable of causing infection outside the xenograft and outside the donor species. There aren't very many specific microbiologic assays, and very few of those are validated for use in humans. Therefore, we don't have a good way of assessing whether we have effective therapies. There's some potential pathogens which look like pathogens that affect humans, such as adenovirus and influenza and coronaviruses and the like. We have some diagnostic tools, but those are pretty much incomplete. In fact, none of those categories has raised as much concern as newer unknown pathogens, the porcine endogenous retrovirus, which my lab cloned, and other organisms that have not previously been identified as human pathogens. As you might expect, since they are unknown, we have neither diagnostic tools nor therapies that are thought to be very effective. Next, please. So the list of potential porcine viruses for which we are unlikely to have complete therapies is actually quite long. Next, please. And what we did years ago, we developed what was called a list of designated pathogen-free miniature swine. What is that? It's a list of organisms that we feel might be effective in causing disease in immunosuppressed humans, for which we need to develop screening assays, and which we would exclude from breeding colonies to make xenotransplantation safer, and at the same time, making the handling of those animals safer for their handlers. Next, please. So some of the early attempts at developing assays, you see Heartline's very nice studies of PCR, multiplex PCR on the right. But one of the areas we need to talk about a little bit is metagenomics, the undirected sequencing. Because if we have unknown pathogens, we might be able to detect them by sequencing DNA or RNA species from the blood of recipients. So you will hear a little bit about not just directed or molecular tests, but undirected molecular tests, as well as serologic testing and others, which may be useful and needed to develop xenotransplantation. Next, please. What are the viruses that we know about? Well, we've studied these for over 20 years. There's the porcine cytomegalovirus, which looks a lot like the CMV of humans. But when it infects an individual with a xenograft, it causes endothelial activation, secretion of tissue factor, with systemic neutropenia and disseminated coagulation and graft rejection. So CMV is a major problem, and you may remember having heard about it in the heart transplant, the xeno heart transplant that was done at the University of Maryland. The porcine lymphotropic herpes viruses cause lymphoma in swine, but have no known effects on human cells. Similarly, porcine circoviruses can cause pneumonitis in swine, but we don't know of disease that would be caused in baboons or humans. The coronaviruses of swine are different than the coronaviruses of human, including SARS-CoV-2, and it's not clear that pigs carry coronaviruses that are infectious for humans, but it is, of course, a concern. I mentioned the PERV, the porcine endogenous retrovirus. There are three types, A, B, and C, and AC is a recombinant form, which is of importance because it replicates better on the only cell line which is used to study this group of viruses. PERV doesn't cause disease in swine, doesn't cause disease in any non-human primates, making them not useful for particular studies of PERV. It has unknown virulence in human hosts, and as I say, doesn't infect human primary cell lines, but only a transformed cell line, which has been used in many studies of porcine endogenous virus. It is susceptible to almost all of the antivirals that were developed for HIV therapies. So should transmission occur, we have the capacity to treat those infections effectively. Next, please. As I say, porcine CMV provokes consumptive coagulopathy in individuals and is resistant to the antiviral agents that we currently have available, which makes it difficult once established to eradicate. So even if it causes infection only of the xenograft, it causes systemic effects, which will adversely affect the recipient, which means that this virus needs to be bred out or excluded from populations that are used as donors. Next, please. The highly replication competent PERV, which we described, are the recombinants between A and C types of the virus and can bind well to human cells. We don't know if they're going to cause any infection in human recipients. As I mentioned, this is a treatable but potentially troublesome infection. Next, please. So the question is, what do we do in the face of unknown pathogens? Next, please. And what do we need to learn? The original effect of cloning of the porcine endogenous retrovirus in 1997 was to ban animal-to-human transplants until we understood more about the various viruses potentially spread from pigs. Next, please. Tom Starzl had a specific take on this, which was that we would have clinical xenotransplantation when I stopped writing papers. Next, please. However, a more direct approach was that we were able to use the new CRISPR-Cas9 based strategy to inactivate PERV originally in a cell line, and then Luhan Yang went on to show that this could be done in pigs that were bred successfully from inactivated viral cells, so that we've been successful in eliminating PERV from certain strains of pigs. Next, please. As a result, and this is from a paper we did in the New England Journal not so very long ago, there are two risks of timeline of infections of post-xenotransplant infections. There are the standard infections that we see in allotransplantation, the cytomegalovirus, the pneumocystis, toxoplasmosis, which are related to the intensive nature of the immunosuppression we use. In addition, there are possible pig-derived pathogens, many of which can be excluded from breeding colonies by careful testing. So the key is having the right assays. Next, please. So we have a designer pig that looks something like this on its designer motorcycle, and we're all able to go ahead and do successful xenotransplantation with the appropriate immunosuppression. Next, please. So I'll stop there, and I'll be happy in the discussion to answer any questions you may have. Thanks very much for including me. I look forward to the rest of the discussion.
Thank you, Dr. Fishman, very much for that outstanding introduction to xenografting and the problem of infectious complications. I'm going to speak next, and let me just share my screen here. Tonight we'll consider protection of xenograft transplant recipients as human research subjects and their competing obligations to others. I have no financial or other conflicts of interest to report. This is a brief outline of my presentation tonight. I'll first discuss the requirements for protection of human subjects in research. Because of the risk of transmission of animal infections, as discussed by Dr. Fishman, I'll discuss whether xenograft recipients must submit to long-term or lifelong surveillance for zoonotic infectious diseases. However, is that ethical? We will review the common rule and other documents that ethically conflict with these surveillance requirements. Others argue that recipients have restricted autonomy and moral obligations to society, and we will explore these arguments, including Ulysses contracts, which are advanced directives waiving the right to withdraw from a research study. We will also discuss alternative solutions to required surveillance. The protections in place for human subjects in research are listed here. First, the safety of the research participant should be the primary concern of the investigator. Informed consent must be obtained that includes the elements of disclosure, understanding, voluntariness, competence, and written consent. There are significant unknown risks to xenografting that will have to be accepted by potential recipients even after precautions have been implemented for their protection. Thus, informed consent will, by necessity, include many unknowns. In addition, how privacy and confidentiality will be approached will be critical because of the expected media interest in any animal to human transplants. Patients will need to be informed about how adverse events will be handled. And finally, investigators will need to be in a state of equipoise when proceeding with xenotransplantation. That is a state of true uncertainty regarding the comparative benefits of each arm in any clinical trial. However, this evening, we're going to focus on one area of informed consent that creates unique conflicts for xenograft recipients: voluntariness. That is, consent must be voluntary, free of coercion, free of any promises of benefits unlikely to result from participation, and the ability to withdraw from the study without penalty at any time. There are several risks that need to be considered when evaluating the advisability of human trials of xenografting that Dr. Fishman has already mentioned. But the issue that concerns us today is the transmission of animal infections into recipients and into society. Because of this risk, especially in light of the recent COVID-19 pandemic, the question has been raised as to whether xenograft recipients must agree to long-term or lifelong surveillance for zoonotic infectious diseases. Because of the risk of interspecies spread of infectious agents through xenotransplantation, thereby putting recipients and society at risk, several groups have made recommendations regarding long-term surveillance of xenograft recipients. In 1999, the FDA banned the use of non-human primates for xenotransplantation due to what was believed to be a high risk of transmitting zoonotic infections. In 2001, the US Public Health Service registered strong opposition to the use of non-human primates and made recommendations regarding surveillance should other animals, such as pigs, be used for xenotransplantation. These included that recipients and their contacts should not donate or exchange body fluids and or parts for use in humans, that they should be required to undergo long-term or lifelong surveillance, even if the xenotransplant is rejected or removed. And finally, the recipient must report immediately any serious or unexplained illness in themselves or contacts. The requirement that contacts report illnesses is predicated on the disclosure by the recipient that they've had a xenotransplant, which could be a violation of their privacy. In 1966, the Nuffield Council on Bioethics wrote that consent for transplant should include an agreement for monitoring. This would include a periodic physical exam and archiving of serum and tissue samples throughout their lifetime. In addition, serum samples from healthcare workers caring for the recipient should be archived. Close contacts should report unexplained illnesses, and recipients should agree to an autopsy at death. The recipient was also supposed to take precautions to minimize transmission of infectious diseases and should not donate blood, tissue, or organs. Dr. Fishman has previously suggested that social and sexual contacts should be monitored if the recipients develop an infection. The recommendations we've just seen, however, are in direct conflict with HHS regulations for the protection of human subject in research that is codified in what is called the Common Rule. The Common Rule defines research as contributing to generalizable knowledge, requires that information be provided to a prospective subject so that they can make an informed decision, and most importantly, includes a statement that participation is voluntary and refusal to participate will involve no penalty or loss of benefits to which the subject is otherwise entitled, and the subject may discontinue participation at any time without similar penalty or loss of benefits. There are similar requirements in the Nuremberg Code, the Declaration of Helsinki, which has undergone several revisions, and the Belmont Report. Dr. L. Sid Johnson, a bioethicist at SUNY Upstate, has written that to require lifelong surveillance of xenograft recipients effectively denies research subjects their fundamental right to withdraw and violates international norms and ethical guidance. Furthermore, she argues that the surveillance and notification of close contacts impinges on the right to privacy and confidentiality of patients and research subjects. Let's consider these competing positions: required surveillance versus the common rule. In bioethics, there are often competing principles when trying to determine what is the right thing to do. Let's consider the premise of the common rule. The common rule is based on the principle of patient autonomy or respect for persons. The competing principles of justice and utility, however, demand a balance between the recipient, who claims autonomy, and the ability to withdraw from a research study at any time, and the rights of society that is being put at risk for transmitted infections by the transplanting of an animal organ. This balance will likely depend on the magnitude of the risk. The greater the risk to society, the greater the moral obligation of the research subject who is the beneficiary of the xenograft to agree to monitoring. However, the risks to the patient and to society are currently unknown, and so establishing what is owed by the patient still cannot be determined. However, the pandemic caused by COVID-19 is a cautionary tale of zoonotic infections spreading rapidly, creating a public health disaster as well as an economic one. The COVID pandemic also raised issues of personal liberty versus public health and which should take precedence. During the COVID pandemic, we debated as a society the ethics, the legality, and the politics of mask, vaccine, and quarantine mandates. Can an individual's autonomy be restricted in a public health crisis? There is actually a legal precedent. Supreme Court of the United States, in a case of a person's refusal to get a required smallpox vaccine during a smallpox outbreak, the court found that the individual liberty to refuse a compulsory vaccine was not absolute, and that an objection can be overridden with a compelling reason, such as the need to protect the public during an outbreak. The court further stated that it did not violate the Due Process Clause of the 14th Amendment. In addition, the CDC has the ability under federal law to restrict freedoms of individuals suspected of carrying communicable diseases across state lines. Finally, states also have quarantine laws that can be enforced. There are some unique issues for pediatric recipients of xenografts because they are unable to consent for themselves. Parents or guardians would have to commit the child to lifelong surveillance. The problem is that it thereby limits their future freedom and opportunities, which could violate the child's right to an open future. There's also a greater chance for non-adherence during adolescence and young adulthood, and this raises the question of whether parents could waive the right to withdraw for their children, and would this extend into adulthood? However, Rousseau states on the social contract that parental rights over their children do not extend into adulthood. In the current pediatric environment, patients would be able to make their own decision about surveillance at the age of assent, 13 or 14 years old, or consent at age 18 and override parental decisions. But let's consider whether waiving the right to withdraw from surveillance or a so-called Ulysses contract can be justified in xenograft recipients. In Homer's poem, The Odyssey, Ulysses had heard the tales of the sirens, mythological beings who lured captains of passing ships toward their island's rocks with their beautiful singing. The ships would crash into the rocks, killing all aboard. Ulysses was on a voyage with his crew and was to sail past the sirens, who he wished to hear sing but wanted to live to tell the story. The goddess Circe told Ulysses to have his crew lash him to the mast, put wax in their ears so they could not hear the sirens singing, and ignore his orders to untie him until they had passed by the sirens and they could no longer be heard. This was essentially an advance directive of his desires during the time he lacked decision-making capacity and while under the influence of the sirens, i.e., a Ulysses contract. The psychiatric advance directive has been classically referred to as a Ulysses contract. It is a legal tool that allows persons with mental illness to indicate their preferences for treatment in advance of a crisis. It is only put in action when a patient loses their decision-making capacity. Ulysses contracts are defined and enforced by state law. The goals of a Ulysses contract include supporting the autonomy of the patient while the patient lacks decision-making capacity. Ulysses contracts, argues Hurst in the reference noted on this slide, are not applicable to xenotransplantation for three reasons that we will consider in turn. First, in a psychiatric Ulysses contract, its implementation depends on the patient losing decision-making capacity. Autonomy is key to the psychiatric use of the Ulysses contract. In xenotransplantation, there is no loss of decision-making capacity. A decision to not comply with monitoring is a conscious, informed decision. Enforcement to honor autonomy is not applicable. Furthermore, overriding the desires of a patient with decision-making capacity to enforce the Ulysses contract would violate their autonomy and their ability to make their own medical decisions. Enforcement would be justified not out of respect for the patient, but to protect society. Second, enforcement of Ulysses contracts in xenotransplant recipients is very problematic. Psychiatric advance directives are regulated by state laws and are not applicable to xenotransplantation. Psychiatric advance directives are enforceable if they meet state law requirements and if the patient does not have decision-making capacity. In xenotransplantation, there is no loss of decision-making capacity for the instruction to take effect. Enforcement would be via contract law, mutual obligations, and public health law, which allows restriction of individual rights. And as I mentioned earlier, this would depend on the risk, the relative risk of transmission of zoonotic infections. Finally, the clinical and regulatory barriers for Ulysses contract in xenotransplantation are too high. Attempts to require long-term monitoring would violate U.S. federal guidelines and the Common Rule and the Belmont Report. Barring exception being made by the HHS and other agencies, attempts to mandate compliance would be opposed by legal challenges. Court or CDC action would likely be a last resort, that is, a threatened pandemic. A possible solution to this inability to enforce follow-up proposed by Hearst is to first better understand the clinical, scientific, and public health value in lifelong monitoring for zoonotic infections in xenograft recipients. In order to increase the odds of compliance, the transplant team will need to provide education and support before, during, and after xenotransplantation. And finally, the transplant team needs to ensure that recipients, family members, contacts, and caregivers understand the potential risk. Hopefully, this results in compliant human subjects. In conclusion, surveillance of xenograft recipients for zoonotic infections places patient autonomy and the rights of society in conflict. Waiving the right to withdraw from a trial or a Ulysses contract is not applicable to xenotransplantation. Legal precedent and federal public health law could compel compliance, but enforcement could be problematic. Patient and family education and support before, during, and after xenotransplantation may improve odds for voluntary compliance with long-term or lifelong surveillance. Thank you very much for your attention. I'd be happy to take any questions during the panel discussion. Our next speaker is Dr. Lisa Moses, who is a veterinarian and bioethicist. She is co-director of the capstone program for the Master of Science in Bioethics program at Harvard Medical School, a part-time lecturer on global health and social medicine, and a member of the HMS Center for Bioethics. She is also a visiting scientist in the vertebrate genomics group at the Broad Institute of MIT and Harvard. Her research, teaching, and writing is split between animal and focused research ethics and veterinary medical ethics. Dr. Moses will speak on animal welfare and animal ethics. Thank you, Dr. Moses.
Thank you, Dr. Hanto. Let me share my screen. Thank you very much for inviting me here. I have no conflicts of interest to disclose. And I want to thank Dr. Fishman for his lecture. It brought back a lot of memories of my swine medicine courses in veterinary school. That was fun. So let me get started. For those of you who are unfamiliar with the satiric newspaper called The Onion, rest assured that this headline is a joke. I'm starting with satire because many of you listening to this talk might be wondering why we are even talking about the pigs or asking about the pigs. And I understand why you might reasonably ask this question. After all, no one disputes that organ scarcity is anything less than tragic for so many patients and their loved ones. So is there any legitimate reason to ask if there are ethical concerns and considerations about non-human animals used for xeno? Well, I actually think that there's some basic issues that deserve deeper attention and some less obvious ethical conflicts that deserve a spotlight. Here's my not so short list of issues and ethical considerations that I've been thinking about in my work and in peer discussions and in teaching about the ethics of xenotransplantation. I want to highlight a couple of basic questions, such as why use animals at all, reasonable animal welfare concerns, but mostly I want to spend a little bit more time talking about what may to this group be the less obvious ethical dilemmas, basically things about animal ethics. And if there are other points that interest you that I don't get to, happy to talk about them in the discussion at the end of this. So why use animals at all? Well, clearly we need big solutions to our organ scarcity. The problem with assuming that xenotransplantation is ethically justified as the answer or the main answer is that we have a long history of justifying almost any animal research on necessity grounds without actually scrutinizing the specific practices that we use. And to me, that's not exactly a robust defense of ethical action. Given that we are using sentient living beings who can suffer in physical and emotional ways, we need moral justification for doing so, since we've decided that this is the exact reason why we can't use human beings in the same way. Regardless of whether or not you feel strongly that animal research is generally fully ethically justified in the service of advancing human health, it's hard to argue that we shouldn't examine the actual practice of animal research to make sure that it's justifiable as performed, not just based on general necessity. One way to think about this is to consider how the practice of using animals as organ sources reflects on human virtues and morality. Does the choice to use animals in this way, both generally and in specific practice, consist of actions that we're comfortable with, even proud of? Or will we look back and think we didn't do enough to ensure that the animals were treated in a way that reflects well on our character as humans? When I talk to people outside of xenotransplant about this issue, it's clear that very few people understand exactly how the pigs are produced for organ procurement. And I think it's important that people realize that these pigs don't have anything resembling a typical life for a pig, even compared to a pig in an industrial food producing setting. These pigs are produced using genetic engineering, cloning, surgical birth, and then they are raised in isolation to keep them pathogen free. They're not raised with a sow, for example. And the cloning process is very inefficient, meaning that it takes a lot of donor pigs, surrogates, and clones to generate a single pig who can produce an organ for xenografting. And this is really a big contrast to how other research pigs are bred and cared for in typical research settings, for example, in common surgical technique research, where pigs are socially housed in welfare-enriched environments. Most of you probably know about the 3 R's framework in animal research. In the US and in many other parts of the world, before animal research protocols are approved, regulations require that attempts be made to reduce the number of animals used or harmed, replace animals with non-animal models, and refine the techniques used to minimize pain and distress. Now, admittedly, this framework is vague and is subject to all kinds of conflicting interpretations. There's legitimate critique that is inadequate to ensure ethical justifiability of a protocol, which is why outside of the US, more robust frameworks are in place. But this is all to say that we need to ask whether or not we're taking reasonable steps to at least try and meet the physical, social, and emotional welfare needs of these pigs. There should be some transparency about those attempts. Let me be frank from a bioethics perspective. The field of xenotransplant research has a history of exaggerated claims of success and a reputation for disregarding animal welfare standards that existed long before the current era of pig-to-human xeno research. For example, years after the UK put a moratorium on xenotransplant clinical trials in 1995, there was a subsequent uncovering of major research misconduct and animal welfare violations, which really didn't help generate trust in the industry. There needs to be a demonstration of willingness to uphold basic standards of responsible research conduct and some real attempts to generate trust on these issues to overcome this. I think it will be crucial for ultimate public acceptance of pig to human xeno. And I don't believe that successful treatment of patients without research integrity is enough. I'm sure all of you have seen information about pigs' intelligence and sentience. When you spend any time around pigs, it's easy to see that they're fully aware of themselves as subjects of their own lives. They aren't like dogs, but they are acutely aware of and interactive with people. This is a picture of a colleague of mine's research on pigs who she taught to use a joystick to move a cursor and play a video game. When they were successful in playing the video game, it caused a food reward to be released in another location. So that means that the pigs understood that their actions had an effect elsewhere and later. The same pigs, after they learned how to use these video games to get food rewards, continued to play the video games for no good reason. So there's no question that we are talking about cognitively and behaviorally complex animals. But like with all human, non-human animal relations, we are full of contradictions and paradoxes in our use of them. We've picked pigs for xenotransplantation for some practical reasons, some of which Dr. Fishman went over, like their comparable size to human organs, but also for their public acceptance of using pigs for other uses that seem less necessary. Plus, there are some cultural values that have led us away from using dogs and non-human primates anymore. Nevertheless, some people find the use of pigs unacceptable. When I explore this idea, what often comes up is a vague sense that we are violating the integrity of the animals in the way we're creating them and using them. It's often hard for people to articulate why this bothers them, but it does. Sometimes it's about general unease with the bioengineering of animals. Sometimes it's about violations of the intrinsic dignity of living beings. And these are hard things to talk about, but they come up all the time and they deserve wider attention. One of the problems with how we regulate animal research use, particularly in the US, is that we only assess whether or not basic welfare standards are met. These are things like ambient environmental parameters and provision of adequate food. We don't actually consider whether the research is ethically justifiable, and we certainly don't consider concepts like dignity or integrity. There's a very big difference in human subject protections and protocol review for animal research. And you may consider that entirely appropriate, but we can't claim that we have such a different standard because animals suffer in fundamentally different ways than people. So can we legitimately ignore public disagreement over using animals for xenotransplantation? I picked out some public comments in response to this recent New York Times report. The first comment here says, is it possible to be simultaneously thrilled for the hope that this brings to those suffering kidney disease and horrified for sentient beings that will be bred and raised in sterile labs only to have their organs harvested? It isn't an either-or situation. Both things can be true at once. So what do we do with the stakeholders and the public who find the entire idea of xenotransplantation repugnant or scary, excuse me, or wrong for whatever reason, even if it's not about animal ethics? I don't feel comfortable with the idea that the desperation of organ scarcity completely overrides dissenting views. I don't think that's acceptable in a pluralistic world, and it's certainly a basic tenet of bioethics. So this is why I want a lot of reassurance that the research is being done as responsibly as possible. Can we find a way to do better with at least the way the animals are created and treated? And let's not forget that thousands of non-human primates are needed to progress with pig-to-human xenotransplantation research and success in the clinical realm. I think that an underappreciated aspect of xeno research ethics is that although much of the public has no qualms about using pigs as sources of organs, they don't feel comfortable with non-human primate research, and they don't realize how many non-human primates are actually involved in this research. Indeed, I would argue that current public acceptance of xeno has a strong foundation in the perception that we are only or mostly using pigs. So how should we think about this? And can we change this paradigm? For example, is the current use of brain-dead humans as research subjects going to allow a reduction in the use of non-human primates in preclinical trials? Are we even thinking about that, whether or not we can use that as a potential way to reduce the numbers of non-human primates? And lastly, I want to address the most common ethical argument for the unchallenged use of pigs for xeno. We are phenomenally successful at producing vast quantities of cheap pork. In the US alone, we produce about 28 billion pounds a year, generating over $8 billion in export value, and that's only about 30% of the total production. And this efficiency and output keeps on rising. But it's generally agreed that this is made possible by disregarding the huge harms done to the health and welfare of both the human laborers and the pigs in industrial food systems. So the argument often stated is that if we are willing to accept that we do this in order to feed people something that they technically don't need to live, doesn't that make harvesting pig organs for people who are dying ethically justifiable? But I really don't understand this logic. It seems to me to say that we cannot question the creation and killing of pigs for organs because of animal welfare concerns, since this is a less bad use than our current practice of pig farming. This argument doesn't say a whole lot about how much less bad xenotransplant is, but it's hard to see that saying something is less bad than a seriously problematic practice is high moral praise. And I think that there's some very interesting philosophical trails to pursue about this logic, which of course are beyond the scope of my few minutes here tonight. So I will refer you to a reference on my last slide from Copland, who delves into the philosophical arguments much more. And of course, we haven't even begun to consider what are the ethical differences between development of the organs as a research endeavor and the production of organ for harvest at scale. These are practically, ethically, and from a regulatory perspective, completely different things. For example, the USDA considers the development of a specific animal species cloning technique as research and regulates it as such. However, once the technique is created and being used for production, it's considered breeding and is no longer regulated. How exactly will this work in xenotransplantation? Some of my work involves analyzing specific research practices and protocols that use animal models. From a research ethics perspective, I look at things like funding ethics and whether private and public research partnerships compromise animal welfare. Using my veterinary training, I evaluate granular details like whether the sourcing of animals is appropriate and whether pain assessment is optimal. What I found when research transparency is enough to allow analysis is that there are often big gaps in meeting even basic regulatory standards. And those are standards that we've agreed are minimums, not best practices. So I think we could go a long way towards improving trust in the current field of xenograft research with some factual assurances that research is being conducted in the best, most ethically sound way. And I think that there needs to be a commitment towards respecting disagreements of values among groups of stakeholders, not just dismissing dissent with a claim that the ends justify the means. And I really think that bioethicists could help make this happen. Thank you. Here are some references, and I look forward to our discussion.
Thank you very much, Dr. Moses. Before we start our panel discussion, I'd like to introduce Dr. Lucy Jefferson, who is a resident in the section of cardiac surgery at the University of Calgary. In 2023, she completed a fellowship in bioethics at the Center for Bioethics at Harvard Medical School and is a member of the Surgical Ethics Working Group. She's particularly interested in the bioethical issues in xenografting and how advances in xenografting will affect the practice of cardiac transplantation. Lucy will just briefly introduce the discussion and raise a few questions that I'm sure that some of the panelists will be interested in. Thank you, Lucy.
Thank you, Dr. Hanto. I appreciate everyone being present here for our session on the considerations of pig to human xenotransplants. As Dr. Hanto said, I'm interested from a surgical perspective in transplantation as a field and in xenotransplantation as a potential niche of this field and in the ethics of it in particular. I will just begin to introduce a couple of discussion questions that are particular to each of our wonderful speakers' topics, but we will address the questions in the Q&A as well. We'll just go to the next slide, please, Ali. Thank you. So in regards to Dr. Fishman's talk, the couple of questions that came to mind for me about the risk of infectious disease transmission in particular include about the specific risks of transmission to the recipient and to the society at large, and whether or not that risk is a static or consistent, constant risk, or if that risk may decrease over time. Is it an acute risk, such as acute graft rejection, or is it constant throughout the life of that graft? What is the risk of severe infection with serious morbidity and mortality? We know that the CMV risk in particular was high for that one donor in the cardiac transplantation. And then how likely would it be that a preventative vaccine or a therapeutic antiviral could be developed that might address and decrease these risks? And similarly, this was a question that also came up, I think, in some of our Q&A of, how many of these questions need to be answered before we would begin to proceed with a clinical trial? And would it be possible to answer these questions beforehand? Some questions that pertain to Dr. Hanto's talk regarding the human research subjects might be that, can we actually require recipients to waive their common rule right to withdraw from a xenograft research study at any time? And would this include after transplantation, and for how long? And that was also a question that came up in our Q&A, I believe. Would lifelong monitoring of transplant recipients be obligatory, and for how long, really, given that potential risk to society at large? And who would regulate this monitoring? And who would collect and possess this data? And a similar question that we did not address per se in this topic, but that might be worth hearing your thoughts on, Dr. Hanto, or any other panelists, would be how long can xenotransplants be trialed in brain-dead recipients and brain-dead patients? We know that is one mechanism in which xenotransplants are being tested. Next slide, please. And then, pardon me, one more question on human research subjects would be, how would we ethically determine who the most appropriate recipient of the xenotransplant be? Would those who receive xenotransplants, would they also be eligible for human organs? Would this be a bridge to human allograft transplant? Or would a xenotransplant be reserved for populations where alternatives such as ventricular assist devices or dialysis might be unavailable or cost prohibitive? This would be eventually. So who would the recipient be ideally? And then along the lines of Dr. Moses's talk, is it overall acceptable to genetically modify pigs with human genes, to alter the DNA of pigs in order to address the human need? And as Dr. Moses was speaking, what is the quality of life for organ source pigs, and how does that compare to the quality of life for pigs bred for other purposes? And do our obligations to pigs that are bred for other purposes, such as in industrial food supply, perhaps change based on our concern for an investment in pigs that are bred for transplant? So if we have a new standard or a standard for pigs based on our appreciation for them as potential organ donors, would that maybe change how we see their importance or how we consider their welfare in other contexts, such as in industrialized farming? So those are just some of the thoughts that I had going through some of these very interesting talks. Obviously, we have some growing questions in our Q&A. So I will turn this back over to Dr. Hanto and the panel. Thank you so much.
If we can have Dr. Fishman and Dr. Moses back on, we have a number of very interesting questions that I'll pose to our panelists. So one of the first questions, and this is a question I think a little bit, Jay, for you and I, at this point, there's a lot of unknowns in xenotransplantation, still at a very investigational stage. Once it becomes a more established therapy, do you think that the requirement for surveillance will be mitigated, will go away, will lessen? Or do you think the risk of latent infections and later transmission will still be an issue, that this will always be a conundrum?
So thanks. It's a very good question. The honest answer is I don't know, but I think one of the things that's happened in allotransplantation, which is the easiest model to look at, is that as we learned more, we actually put on different levels of surveillance for our patients over the past 40 years. And I think the same would happen with xenotransplantation, which is that we need to learn from the early patients and then move on. So for example, from the heart transplant at the University of Maryland, they became aware that serologic testing, antibody-based testing, was necessary for porcine cytomegalovirus because you needed to be able to screen the animals using serology, not using molecular testing. So the nature and the duration of the screening and surveillance we do will depend on the outcomes and our experience. So we do the best we can. And the FDA, for example, is very strict, both in terms of the animal rearing and the clinical trials. And then we have the IRBs as well. But it's very important that we recognize that this is going to be a learning process, much as it was for allotransplantation.
Well, maybe I'll just tag on there, Jay, another question that's somewhat related, and that is, what do you believe the metrics are going to be for starting a clinical trial? Because as you've pointed out, some of this is still quite unknown. Do you feel that, you mentioned that there are some therapies for some of these viruses and there is a certain amount of testing. Do you feel that from an infectious disease standpoint that it is safe to proceed? Do we know enough? Do we have enough tools in our quiver to pursue this at this point?
So one of the advantages of having corporate sponsors of the pig breeding and the potential sources of animals is that they are used to and are tightly regulated by the Food and Drug Administration. The advantage of that is they need to come forward with trials under FDA guidance, which exist, for screening of pigs, screening of humans, and then they go to institutional IRBs. Do we know how safe it is? We don't. But in general, we've been unable to infect human cells in vitro or baboons in vivo with pig pathogens. And the exceptions being something like porcine cytomegalovirus that causes disease, even though it only infects the xenograft. So we need to learn some more, but the regulatory environment is very strict right now, so that we're archiving baseline samples for testing should something occur downstream. How long will we have to do that? Don't really know, but they're saying 20 years minimum. So it's going to be a long time. It really falls, Doug, into your area of thinking about whether a patient can opt out once they've opted in to getting a transplant because they are a potential source of infection for the community.
Right. Well, I think as I talked about, that is, I think, in part going to be determined by the nature of the risk. So if there is a risk of significant transmission into society and the creation of a pandemic, then we're probably in a better ethical situation of requiring surveillance than we are if it's, an unlikely event.
I think it's an unlikely event. I think pandemic is very unlikely. I think the potential for infection is always there, but I would be more surprised if they didn't get a lot of infections similar to the ones that we see in allotransplantation that are derived from the recipient and the community. So I think the safety is pretty well established. I do think that we're not going to be able to definitively answer those questions in the absence of clinical trials.
Great. Thank you, Dr. Fishman. Dr. Moses, there's a question about whether there is a metric to assess animal models' levels of pain. I think they're referring probably specifically to pigs. And could such a system be helpful in the ethical treatment of pigs being used for xenografting?
Oh, sure. There is a system in place for pain assessment of animals used as laboratory research subjects, and that is part of every approved protocol in a research setting. So there's lots and lots of metrics and guidelines for assessing pain. The problem in typical animal research settings, as opposed to what's happening in xenografting right now, where we're dealing with very small numbers of individual pigs, is most of the time when these things are done at scale, it is very difficult to treat those research subjects as individual patients. There is some move afoot in some places to treat animal research subjects as individual patients and assess their individual pain. But, and we certainly have the ways to do that. That's what I do. But in typical situations where you may have something like 300,000 mice in a facility, it's quite difficult to assess individual animals' pain and act on it.
Okay, thank you. Here's a question that as access to xenotransplant expands, the question will come up as to whether or not xenotransplantation is a right or whether it is something different. So, especially in the context, Dr. Fishman, where we were talking about long-term monitoring. And can you imagine a situation in which such access, either in research or clinical setting, is a right, either now or in the future, and what that would mean for long-term monitoring?
I think there are two ways to approach this question. I think it's a great question. First is in the context of allotransplantation, where we generally agree we have a limited resource of human organs, but it is a right for everyone to be considered for an organ, and that they should be without regard to ability to pay or race or religion or any other criteria. So that It is a therapy for end-stage organ disease, which should be available to everyone. But clearly it's not. And that's why we have stewardship of organs, because of the fact that we just don't have enough. And that's why xenotransplantation was even up for consideration. So we need to be sure that both in clinical trials and in clinical application, once this is in clinical practice, that everyone has access, equal access to these organs. Will we have enough to start? Probably not. Will they be expensive? Well, they'll largely be borne by corporate entities before the insurance companies or Medicare will pay for it. So that I think it's an opportunity to make sure that both in clinical trials and in application, that everyone has equal access. The difficulties being financial largely, and that there are going to be a smaller number of centers that have the capacity to use these type of organs.
Dr. Fishman, let me just tag on then. There was another question about actual allocation of xenografts. Once xenografts are more available, how do you believe they will be allocated? Will it be similar to the system that we have now for allotransplantation?
Honest answer, again, don't know. I think that it's important to discuss who the initial subjects will be. And the initial subjects might be people who, for one reason or another, couldn't get an organ, a human organ, or that had urgency for an organ that wasn't available to them at the time. There are many, and we've written a little bit about this, but so that I think that the key is what the relationship of xenotransplantation is to the allotransplant system. For example, if somebody is in a xenotransplant trial, they shouldn't lose their place in line for a human organ should that xenograft fail. So that there are a number of complexities in the interaction between allotransplantation and xenotransplantation. But I don't think anybody has any idea what large-scale xenotransplantation is going to look like.
Okay, thank you. Dr. Moses, there's a question here about what is your intuition regarding why pigs are socially acceptable sources of organs as opposed to non-human primates? And perhaps more importantly, does it really matter?
I think this is an excellent example of the contradictions that we human beings have in our ideas of what animals are and what they should be to us. You know, I think this argument that comes up every time we talk about this, that since we eat so many of them, why can't we do this, is a very good explanation for that. You know, I think that maybe more telling is not the only, you know, we, as Dr. Fishman mentioned, we stopped using non-human primates organs for xenotransplant research a long time ago. And of course, a lot of that is for zoonotic concerns, but there's also very much cultural and ethical reasons why it is largely not acceptable to lots of people to use non-human primates. And I think the fact that pigs have been chosen is not just because of all the biologic reasons that we know of that make them suitable for this. I think it's also because lots of people think it's just okay to do this. So I think we have to be clear that, yes, we're talking about research and we're talking about clinical practice, but of course there are cultural and social contexts to all of the decisions that we make. And I think the choice to use pigs very much fits into that definition. I did want to actually come back to something that Dr. Jefferson said, which I thought was really intriguing and I hadn't really thought about, which was if pig to human xenografting becomes successful, might that change the status of pigs in our culture? And I think that is a fascinating thing to consider, particularly because we have religious and cultural groups of people who do not eat pigs for a variety of reasons. And there is certainly in some cultures a lot of feeling that pigs are unclean and not deserving of welfare considerations. So we have to sort of square away all of those paradoxes and contradictions. And I think this idea that perhaps it would change the status of pigs and maybe prompt us to think again about the way we treat them in industrial food systems is fascinating, and I would love to explore that more.
Great, thanks, Dr. Moses. There were some questions specifically about that issue. We obviously have an international audience, and pigs are viewed differently in different parts of the world. So thank you very much for that answer. One little short question that came up for anyone is, were there similar concerns raised by the use of pig valves for human hearts?
Not from an infectious disease perspective. I can't speak to the ethics thereof, but the pig hearts are generally formalin fixed, which we assume will kill any organisms that are present. But I can't address the question of whether or not. One of the interesting features is that we've just learned in the last decade or so is that one of the reasons pig valves don't last longer is they still have the alpha-1,3 gal sugar on their surface. And so they degrade by the same immune mechanism that causes hyperacute rejection of xenografts.
Dr. Moses, any comments just about the use of pigs? Because this has obviously been going on for quite a while, but a little bit under the radar, in fact.
Yeah, I think the fact that this practice became so widespread such a long time ago, before there was more discussion about animal ethics, probably it's almost grandfathered in as socially acceptable in a way. But yes, there have definitely been people who have thought about that and written about it. I think, though, it doesn't provoke the same kind of controversy and feelings as the newer uses where we're actually doing all of the bioengineering. I think that is a line for some people that really has sort of changed this conversation.
Great, thanks. Dr. Moses, there's also a question, again, related to the value and how we view pigs. Slightly different, though, in the sense that this refers to your comment about them being knowledgeable about their life. And there is a question, I think they would like you to expand on what that means and where does that place them along the sentience metacognition spectrum, and should this matter for their moral status and for their use, or how they're treated if they are to be used as xenografts?
Well, I think the use of animals as research subjects in general, most of the time people tend to sort of think of this in hierarchy, and they ascribe different categories to different kinds of animals. And for many, many people, I think it's pretty common for people to feel like animals who seem closer to human beings, they feel less comfortable using them in research settings, which is why I think there are far more people who do not feel comfortable with the use of non-human primates in research, but are okay with other species. But conversely, there's lots and lots and lots of people who are not comfortable with the use of dogs as research subjects, and dogs are actually quite commonly used as research subjects. And that's clearly not specifically about the dogs themselves. It's about the relationship that human beings, the truly unique relationship that human beings have with dogs. So there's a lot to this. What I was saying about pigs understanding that they are the subject of their own lives. Pigs are one of many species of non-human animals who have done things like pass the mirror test, which is a test that is used in people and also in many species of animals to see whether or not they recognize that they are seeing their own reflection in a mirror, and that a spot that is on their face or their body that they are seeing in the mirror, do they understand that they have to touch their face to touch that spot, or do they touch the mirror? And pigs absolutely pass the mirror test. They do understand that they are looking at a reflection of themselves and that they are themselves. So I think you have to take that as you will. And again, all this comes down to whether or not you think we have a necessity claim simply because we are all part of the human species.
Great. Thanks, Dr. Moses. There is one question here about even if we get recipients to waive the rights to withdraw from lifelong monitoring in terms of logistics and feasibility, how would it be enforced? And I think that is clearly the problem. After allotransplantation as well, and Dr. Fishman has mentioned this, that we have difficulties with patients being compliant with taking their immunosuppression, making clinic visits, coming in for biopsies, just as we will probably have with xenografting. And it begins to fall to the patient-physician relationship. And I think it's going to be very important for physicians, nurses, coordinators, and others involved in the care of the xenograft recipient to try to help them understand the importance of ongoing monitoring, and that this is not only good for them, but good for society, and maybe a little less emphasis on the obligation, but that this is something that is something that will be beneficial to all, and hopefully we'll have more people consenting to long-term surveillance than those becoming non-compliant. Dr. Fishman, there's a question here about xenotransplantation acting as a bridge to allotransplantation. And what are your thoughts about xenografting, let's say early on versus later? You did mention that some of the recipients may be chosen because they're not a candidate for allotransplantation. But let's say that some of them were potentially candidates. Would this be seen as a bridge to allotransplantation at some point?
It's completely possible. I think if you were going to pick an organ that would be most likely as a bridge, it would be the liver, because we see a lot of patients who need to be stabilized clinically with liver failure before they could tolerate a long-term human allograft. It is possible then you could hook somebody up to a pig liver, because we don't have a dialysis machine type device that works for liver replacement. So that's one possibility. I don't know that anybody would want to do that as a uniform approach to xenotransplantation. What the hope is that in the realm of doing no harm, I think that you want to put in an organ that will work for the longest possible time to augment the list, because the current challenge is a shortage of organs. And if it's a bridge to transplant, it doesn't really address the question of where do we get more organs for human transplantation.
Great. Okay, well, we are past our bewitching hour. I will take one other just short, not an easy question, but that just came up here at the end. A question about for children under the age of assent and for religious and cultural reasons, family refuses xenotransplantation. Who advocates for the child? This is one where I wish we had Dr. Jonathan Marin on our panel to delve into this question. I bring up the question because I think it is a really important question. But again, as Dr. Fishman has pointed out, many of the questions that are going to come up with xenotransplantation have come up with allotransplantation. So we have all seen situations where a child has end-stage liver failure, particularly now where we have a much more diverse population in the United States with different religious and cultural views, where families want everything done short of a transplant. And ultimately, in those settings, it does become a family decision, particularly for the very young child. But it again becomes incumbent upon the team to try to educate the family, try to address their cultural concerns, meet with other members of their community, try to understand what their objections are, and see if there is a way to address them. Dr. Fishman or Dr. Moses, do you have any thoughts about that?
My only comment is that some people are advocating that some of the early uses of xenotransplanted hearts would be in children with congenital anomalies, and where there might not be options in terms of human hearts for those young children. So that it's certainly worth thinking about, I think, but there always are protections built in that are unique for children in any clinical trial and in clinical care. So I think they will be well protected and well represented, but exactly how organs will be allocated, I don't quite know.
Okay. Well, great. Well, this brings us to the end. I would like to thank our presenters tonight, Dr. Fishman, Dr. Moses, and Dr. Jefferson for their outstanding and provocative talks and discussion of ethical considerations in pig to human xenotransplantation. I'd also like to thank all of you who listened tonight and asked questions of our panel. It was a pleasure to moderate such an interesting discussion. I would also like to thank Dr. Robert Trug, immediate past director of the Center for Bioethics, for his support, and Dr. Rebecca Brandell, the incoming director of the Center of Bioethics, for her continued unwavering support of this conference. Finally, I would like to thank Ali Stitham, Melissa Nosel, and Kyle Amato for their technical, graphic, and organizational support of these conferences. On behalf of the Surgical Ethics Working Group, we thank you for attending. We hope that you have enjoyed and learned from tonight's presentations. We hope that there is information that you will be able to use to address bioethical issues in the future. I do apologize for some of the questions we didn't get to, but know that we will be considering those as we continue our discussions. Thank you once again, and good night.